Welcome to the fascinating world of histology, where tissues tell stories and diagnosis begins at the microscopic level. Have you ever wondered what happens to a tissue sample after biopsy? In this video, we'll walk you through what really happens inside a histology lab, from the moment a tissue sample arrives to the moment it becomes stained, mounted slide ready for microscopic analysis. Whether you're a medical student, a lab technician in training, or just curious, this is your behind-the-scenes science of cells and structure. Every histology journey begins with a specimen, often a biopsy or surgical tissue. Once it arrives in the lab, the first step is accessioning. That means assigning a unique identification number to the sample, logging it into the lab information system, and verifying patient details. This identification will follow the tissue through every step, ensuring traceability and accuracy. Next comes grossing, the macroscopic examination of the tissue. A trained pathologist or histotechnologist describes the sample size, color, texture, and any visible abnormalities. Then, they trim it into smaller pieces that will fit into a tissue cassette. A tissue cassette is a small plastic container that holds the sample during processing. By the way, careful trimming ensures pathologists get the clearest picture possible for accurate diagnosis. Next up is fixation. To preserve tissue structure and prevent decay by autolysis or putrefaction, samples are immersed in a fixative, which is usually 10% neutral buffered formalin. Fixation preserves delicate structures, making sure nothing is lost before analysis. The duration of fixation is critical, whereby underfixed tissue may show artifacts, while overfixation can mask antigen. Now, the sample takes a chemical journey. It is first dehydrated with ascending grades of alcohol, that is, from 70% to 95%, then to absolute alcohol, then cleared with xylene and infiltrated with molten paraffin wax. This process takes several hours, often overnight, using a tissue processor machine that cycles the cassettes through a series of reagents. Tissue processing makes the tissue firm enough to slice into ultra-thin sections. Therefore, without it, the tissue will be too soft and fragile to handle. Once processed, the tissue is embedded in a block of paraffin wax. This step is all about orientation. The way the tissue is positioned affects what's visible and the final slide. The cassette becomes the mold and the molten wax poured in. Once cooled, the block is ready for sectioning. Now comes the art of microtomy, cutting ultra-thin slices of tissue using a microtome. Each section is about 3 to 5 microns, which is thinner than a human hair. The ribbons of paraffin are floated on a warm water bath and then picked up on a glass slide. Tissues are colorless, so we stain them to reveal structure. The most common stain is hematoxylin and eosin, whereby hematoxylin stains nuclei blue, while eosin stains cytoplasm and everything else pink. Other special stains such as MGG, methylene blue, and GEMSA, or immunohistochemistry, can highlight bacteria, cancerous cells, or even rare diseases. Therefore, staining reveals structures and abnormalities in invisible plain tissue. Last but not least is mounting. After staining the tissue, slides are mounted with a cover slip using DPX. This protects the tissue and allows for long-term storage. The final slide is then labeled and ready for microscopic review. Now the pathologist takes over. Using a microscope, they examine the slide for cellular architecture, abnormalities, and disease patterns. They may request deeper sections, special stains, or immunohistochemistry for further clarity. Once the diagnosis is made, the pathologist generates a report, which is often integrated into the hospital electronic medical report. Slides and blocks are then archived for years in case of future review or legal reference. And that is the journey. From tissue to diagnosis, histology labs are the silent engines of modern medicine, turning tiny samples into life-saving insights. If you are entering this field, know that your precision, patience, and passion will shape the future of diagnostics. Thanks for watching. Next time you hear a biopsy, you'll know the incredible journey behind the scenes. So, like, subscribe, and drop your comment what lab process you want to see next.